Hi, this is a material presented by the Canadian Bioinformatics Workshop, and this lecture is presented under a Creative Commons license. You have the right to use and modify the materials at the condition that you indicate the source of the information and the changes that you made. Today, we will talk about gene function prediction and a tool called GeneMania, and it allows to do gene function prediction. So on this slide, beside the workshop logo on the left, we can see on the right the GeneMania logo as well as the GeneMania network. We will see two use cases. During this lecture, we will review some concepts important to gene function prediction. So where are we in our learning path for pathway and network analysis? During this workshop, we see two ways to use pathway database. The first one is to use pathway database in pathway enrichment analysis. But we also can use this pathway database to build functional interaction networks, and where genes would be the nodes and they are linked together if they are functionally related. And once the network is created, and this network can be quite large, we can apply network analysis methods to unbiasedly analyze the network. And for example, we can cluster this network into some modules. And today, as step number 4, we go one step further, and we use all different types of functional interaction networks that we integrate together, and we will use them to predict gene function or to expand our gene list using related genes. So this is the list of our learning objective modules of this lecture. So be able to use a gene recommender system, GeneMania, to answer two types of questions about gene function. First question is, what does my gene do? And the second question is, give me more genes like this. So we will go over the concept of gene function prediction, which are the functional interaction networks, gene recommender systems, guild by association, label propagation, context-specific networks using weights. And then I hope that at the end of this lecture, you will be able to understand how to use GeneMania to interpret a gene list derived from your omics experiment. So in the outline, we will first start by the functional interaction network. Then we will go over the concepts in gene function prediction, covering the gene recommender systems. Then we will explain GeneMania, so explain the network weight system and the scoring interaction by guild by association. Then we will see that GeneMania is available through a web app or a cytoscape app. We will see some application of GeneMania, and finally we will list two other related apps, String and FunCop. So functional interaction networks, how to use large genome-wide data to construct functional networks. So over the past years, the vast amount of large-scale mixed data have been generated and are now stored in various databases. How to make use of the large amount of information that they contain. These data sets provide valuable insight and can be integrated into functional networks for deeper analysis. These various databases for omics data can be categorized based on the type of data that they store and their primary focus. So they can be, for example, genomics database that store genome sequence annotation. They can be transcriptomics database, like the one stored in GeoProteomics database that include protein structure or interaction data, or pathway and network database that store pathway or gene regulatory networks in interaction. So on the slide from left to right, we can see illustrated the transcriptomics data, which tell us how much a gene is expressed in some specific condition or tissue type. And then we have the genetic interaction data. So in genetic interaction data, the two nodes are the genes, and they are linked by a gene if there's a genetic interaction. So for example, the genetic interaction could be the additive effect of the mutation of both genes, and so could represent synthetic lethality. Then we see the protein domain similarity networks. So the two genes here, the two proteins, are linked by an age if they share a common protein domain. Then we have the protein-protein interaction domain. So the two nodes are linked if the two proteins are known to physically interact with each other. And then we have the pathway interaction network. So the genes are connected if they belong to the same signaling pathway, for example. So the question today is illustrated by this little cartoon representing a researcher. And this question is, can we combine all of these functional interaction networks in a useful way and use this information of well-known genes that are in these functional interaction networks to get more knowledge about uncharacterized genes that are also in these functional interaction networks? Indeed, most successful computational approaches for protein function prediction integrate multiple genomics and proteomics data sources to make inferences about the function of unknown protein. So genes with similar patterns of expression, synthetic lethality, or chemical sensitivity often have similar functions. Additionally, functions tend to be shared among genes whose genes product interact physically, are part of the same complex, or have similar 3D structure. So the first step is to construct this functional interaction network. So here we can see the example of a co-expression network based on transcriptomics data. So we see on the left the matrix of all genes and in all conditions, and correlation is calculated to retrieve the genes that have an expression that go up or down in the same way through the different experimental conditions. So then the network is created, and then we see the network on the right is the resulting co-expression functional interaction network, where the nodes are the genes, and they are linked by an H if they were found correlated in the transcriptomics database. We can see that the genes have different widths. It represents the weight. So the width of the H represents the strength of the correlation, and we give more weight to a pair of genes with strong correlation. So there are varieties of functional interaction networks. So some can directly measure interactions, like the one that we've seen, so the protein interaction network, the gene interaction network. Some can be inferred interaction from a single data source. So this is the example of the co-expression network that we just have seen. Some are inferred interaction from multiple data sources, and in a context independent, like the FI network or the string network that we'll see at the end. And sometimes this inferred interaction for multiple data sources can be context dependent, and that's what we are going to see with Gene Mania. So when we have these networks, what are we going to do with it? We will use it to answer two types of questions. What does my gene do is the first question, and give me more genes like this is the second question. So for the first question, what does my gene do? I have one gene. I will try to determine the function of the gene based on who it interacts with using the guild by association concept. For the second question, give me more genes like this. How to find more genes in the Wnt signaling pathway, for example. So we will look at the network that relate these genes together, that will provide some context on how to expand and find more genes like my query list. For example, so if the genes are in the Wnt signaling pathway and all these genes maybe are kinases, maybe it makes sense that the other genes like this and that we will add to the network will be kinases as well. So Gene Mania is a gene recommender system based on functional network. So what is a gene recommender system? So a gene recommender system helps identify genes related to a specific biological function, disease, or process based on data from known relationships. Therefore, we can understand that heavily relies on the functional networks that will be used for this recommender system. So it's like how a movie recommender suggests movies that we might like based on our preferences, and when we use it with our gene list, it will help to add related genes to it. And more globally, it helps to understand complex biological systems more efficiently. So now, so we will go away from the biology just to understand what is a context-dependent query. And so we will use a well-known recommender system, which is ChatGPT, to find 3 other cities like Memphis. So Memphis, the city in Tennessee, US, and then we will provide two different contexts. So the first context, we will add 2 other cities to Memphis, which are Knoxville and Nashville in Tennessee as well. And for the other ones, the query represents a different context, and we will add Alexandria and Cairo to the query. So we will use ChatGPT, which is also a context-dependent recommender, but that use large-scale language data to predict the next word. So let's see what's comebacks. So when the query is find 3 cities like Memphis, Knoxville, Nashville, which are 3 cities of Tennessee, then we get back Austin, Louisville, and Atlanta. And then when we see the description of the city, and you see the word highlighted in yellow, when we see music and culture. So the things that were in common between these 3 cities were music and culture. So that's why ChatGPT added 3 other cities that were also rich in music and culture. Now, when we see the other queries, Memphis, Alexandria, and Cairo, ChatGPT gave us back Babylon, Lagos, and Paris. And when you look at the definition of the city, you see that the keywords are ancient, historical, and culture. So it means that in Memphis, Alexandria, and Cairo, the things in common were also ancient and cultural. That's why ChatGPT gave us back the cities that also had these keywords. So that was an example of how a recommender system works using context-dependent query. So how does it work in biology? So gene recommender for biology, that could be, for example, to find genes related to P53. But that question could be about its biological process, its biochemical molecular function, its cellular localization, its role in disease. So some networks may be better than others for some types of gene function. So when we query just one gene, there is no context. This query will use all the networks equally and add a related genes to our query. But for the second question, give me more genes like this, we include TP53 in the other list of genes. So the gene recommender system is going to create first a functional interaction network with all the genes in the list to understand the functional interaction networks that are important for the other genes in the list to provide a context. And based on the context, so based on this functional interaction network that have more weight in the query list, then the gene recommender system is going to add other genes related to my query, and then they will be different from the query where we only put TP53. In summary, in a context-dependent query, we will choose what networks are relevant to answer the question about that list. So what did we learn in part 1? We reviewed the concept of functional interaction network, gene recommender system in the context of gene function prediction.